Hello students, welcome to Distinction Learning Platform. In this video, we'll be looking at the loop of Henle under renal physiology. But before we begin our lessons, make sure that you subscribe, like the video, as well as comment in the comment section. Alright, let us begin. Now, what we'll do here is that we're going to look at first the anatomy of the loop of Henle so that it helps us to understand the complex processes that will be happening in the loop of Henle. But just to recap of whatever we talked about in two previous videos, we talked about two important things. We talked about the glomerular filtration as well as the processes that happens in the proximal convoluted tuber. And we said to say this A, which is the efferent arteriole, okay, this efferent arteriole is going to bring blood toward the glomerulus, okay, and this is the efferent arteriole, okay. This one is bringing blood toward the glomerulus there, okay. And what's going to happen to that blood? Basically, in blood we're going to have what we call plasma, and in plasma we've got the smaller substances that are dissolved there, and they are very, very small, whereby they can pass through this glomerular filtration barrier, okay. So they are able to pass through here and enter into this Bowman's space. This is what we call the Bowman space. So everything is coming here, and this whole process is what we call glomerular filtration. That's what we talked about in that first video. Make sure that you watch about that in details. All right. Now, since we have this fluid which is inside this Bowman space, and we call it the filtrate, and this filtrate is going to continue the journey into the next segment of the nephron, which we call the proximal convoluted tuber. The proximal convoluted tuber is going to have two important processes that we need to remember. All right. We talked about the tubular reabsorption, where this filtrate, okay, where this filtrate, which is here, let's just say filtrate, which is F, which is here, is going to be reabsorbed back into the what? The bloodstream. That's what we call tubular reabsorption. And this part is what we call the proximal convoluted tuber. This is the part of the nephron, okay? That's the first process that's going to happen. The second process is where now this blood vessel, which is near to the proximal convoluted tuber, will start secreting substances that were not filtered on this glomerular filtration barrier. It's going to start secreting those substances into the tubular lumen, and that's what we call tubular secretion. We had a good example of that, which we said penicillin, which is a drug, okay? Drugs that are going to be secreted in the tubular lumen. Remember about that, okay? Now, after the proximal convoluted tuber, there is this part now, okay? There is this part starting from here, the one that goes down and the one that will go up. It's more like forming a U, okay? So meaning that that structure is what we call the loop of Henle, okay, or the loop of Henle. So the U-shaped structure part of the nephron is what we call the loop of Henle, okay? So meaning that the loop of Henle, let's talk about the anatomy of the loop of Henle, okay? Since we reached this part. So now we're going to say the loop of Henle basically is going to have two important parts. The part that is going downward and the part that is going upward. So the part that is going downward, we call it descending limb, okay? The part that is going downward is what we call the descending limb. And this part which is going upward is what we call ascending limb, okay? Now, I need you to make sure that you understand something here. On the descending limb, it's just an overview concept that we need to take note, okay? On the descending limb, here, water is permeable. This is a very, very important concept that you need to understand only. So let us put only. So on this part that is going downward, okay, that's where water is permeable only. What does it mean? It means that other solutes like ions, sodium, magnesium, they are impermeable. They can't come out of this tubular lumen or this descending limb of the nephron. Okay, basically this, they can't come out and go into the interstitium or into the blood vessel. They can't come out because they are impermeable to that. You need to remember about that. Then on the ascending limb is more like the opposite. Okay, water here is impermeable. Okay, so we are saying here on the descending limb, water, so let's say H2O, is impermeable. Okay, and permeable to what? To solutes like what? Sodium chloride. So let's just say sodium chloride, because those are the important components or ions that are important in the what? Ascending limb there. Okay, so we are simply saying that in the descending limb, water is permeable only, but solutes or ions are impermeable. Okay, then in the ascending limb, water is impermeable, but it is permeable to sodium chloride or other ions like magnesium, potassium. They are also permeable in the what? Ascending limb. That is a very, very basic idea that you need to understand before you go to the complex processes. Okay, now apart from understanding about that, we need to answer a very important question here. Okay, we need to answer a very important question. Why are we talking about the loop of Henle? Because before you dive into those complex processes, we need to understand why the loop of Henle is very, very important. Okay, so the loop of Henle, it is very important because, so this is a very important point that you need to take note. Okay, the loop of Henle is very important when it comes to what? Two important conditions, because loop of Henle is going to make sure that it solves two physiological problems that the kidneys is going to face. Okay, so it's going to make sure that it solves this problem, which is the first problem is what? When the body is experiencing what? High water content. Okay, high water content or high body water content. When you have lot of body fluid, or basically you have lot of water in the body, okay, what's going to happen to the kidneys? Okay, another important question that this loop of Henle, they are going to help to answer or to solve is when you have what? Low water content. Okay, high water content and low water content, these are two important physiological conditions that are going to be happening in your body. Either you have excessive water or you have low water in the body. Okay, now we know that if you have high water content in the body, the kidneys or the nephrons, they are going to excrete what? Dilute urine. It simply means that you are going to excrete more water in the urine. So meaning that on this point, we are going to have dilute urine. Okay, we are going to have dilute urine. Why? Because you have high water content in the body. So the kidney, they are going to make sure that they excrete more water from the body. Okay, no wonder you are going to have dilute urine. Dilute means that you have got more water in urine. Okay, but low water content, basically the kidneys, they are going to make sure that they excrete what we call concentrated urine. Okay, concentrated urine. Now remember what I said. These two important physiological problems, the loop of Henle, they are going to make sure that the kidneys, they are going to what? To solve these two important questions. Okay, two important problems. They are going to help in maintenance. If you have high water content in the body, the loop of Henle are going to make sure that they provide anything that's going to make sure that you have dilute urine in the body. Okay, and if you have low water content, it's going to make sure that it's going to provide the condition necessary to concentrate what? Urine. It's because of what? The loop of Henle. You need to understand. So these ones. So take note of this. Before we start anything, make sure that you take note of this. Alright? Now, since we know about this, now let's go to what happens here. Okay? What happens to this part now? So let's go to this part. So what happens to this part? Now, this part is going to have lot of structures. So now let us understand the important structures that we are going to be using in the loop of Henle. Okay, so we've got this part. This part is what we call the descending limb of the what? Loop of Henle, and it's going to have specialized cells. Okay, this part is going to have what? Specialized cells, which we call epithelial cells. So this part is going to have epithelial cells lining this part. Okay, so let us say epithelial cells lining this part, this part. So these are specialized epithelial cells. Also this side we are going to have specialized epithelial cells. Okay, now what about this part? This part, what do we call this part? This part is what we call the we call it the interstitium. Okay, this part, or this part, as long as we're not talking about inside the loop of Henle, basically the outside part is what we call the interstitium. Now there are two important interstitium that you need to understand. There is the interstitium that can be found within the cortex, and there is an interstitium that can be found within the medulla. So meaning that we're going to have medullary interstitium as well as cortical interstitium. Okay, so meaning that if you look at the division here, if you look at the division on this part, okay, this is where the proximal convoluted tubule is ending. Okay, that's what we call, this region is what we call the cortex. Okay, where the proximal convoluted tubule is ending, that's where the cortex is going to end as well. Okay, but where these extension, these loops of Henle are starting from and extending down, we're going to have what we call the medulla. Okay, this is the medulla. Simply means that if this is the medulla, meaning that this space is what we call medullary interstitium. What about this space? This space we call it cortical interstitium. Okay, why am I troubling you with the interstitium? Because something happens in the interstitium of the loop of what? Henle. Okay, make sure that you understand. Now let us understand something here. Since we know that this is the medullary interstitium, okay, this is the medullary interstitium. Now in the medullary interstitium, there is something that's going to happen. There are some numbers that I'm going to be putting here. Now, before I put those numbers, we need to understand a certain concept that's going to help us understand everything now. So let us understand about this concept, which we call osmolarity. Okay, what is the important concept that we need to understand? Osmolarity. Okay, so we need to understand about this part, which is osmolarity. Okay, osmolarity, the one with L. So what is this osmolarity? So osmolarity now talks about the amount of osmotically active particles that are present in what? 1 kilogram of water. Okay, we use kg in osmolarity. Okay, we use kg in osmolarity. So meaning that osmolarity, if you look at the if you look at the units for osmolarity, you find out to say we're going to have these units. Okay, so let me just use blue. We're going to have what we call mosmol. Okay, you're going to find something like this, mos per kg, like this. Okay, you're going to find something like this. So meaning that, as you can see, as long as you are able to see that the denominator is having kilograms, that's what we call osmolarity. But if you talk about something like having the units like mosmol, okay, per liter. So if you put liter here, then you are talking about osmolarity, the one with R. Okay, so you need to take note of that. Now, in physiology, most of the time we use the term osmolarity. Now, what is osmolarity again? We said osmolarity basically talks about the number of osmotically active particles that are present in the 1 kg of solvent or water. Okay. Now, what do I mean about that definition? What does it mean? So it means that if you talk about osmotically active particles, basically you are talking about ions. So ions that are osmotically active, meaning that ions that are able to drag water. The moment they move from one compartment to another, they are able to drag water with them. So mean that those particles are osmotically active, because there are other ions or molecules that are not osmotically active. Let me give an example like bicarbonate. Bicarbonate is not osmotically active. Reason being because the movement of bicarbonate doesn't drag water, so it's not osmotically active. Okay, so we need to know about that. Now, since I've troubled you about the osmolarity, now what is the normal osmolarity of the proximal convoluted tubule as well as in the Bowman space? What is the value that's going to be here? Because osmolarity, we're talking about the number of particles present in in the 1 kg of 1 kg of water. So here, the plasma that was filtered here, it has a certain osmolarity similar to the blood vessel, or similar to the serum there. Okay, so we're going to have about, in the Bowman space here, we're going to have about 300, okay, more, okay, per kg, like that. So we're going to have 300 here in the Bowman space. But if you go to the proximal convoluted tubule, remember what I said, there will be reabsorption of some ions there. But remember also water is going to be reabsorbed. So if water is also reabsorbed back into the bloodstream, as well as the ions like sodium that are reabsorbed back into the bloodstream, the osmolarity is not going to change. So the concept is that from the Bowman space up to the proximal convoluted tubule, the osmolarity doesn't change. So normally, under physiological conditions, okay, normally this is not going to change under physiological conditions. But under some certain circumstances, maybe if you have metabolic acidosis and maybe some other conditions, this value is going to change in the proximal convoluted tubule as well as in the Bowman's space. All right. Okay. Now let's talk about this part. So this part, we say this is the descending limb, but basically this is what we call the thin descending limb. What's going to happen to this osmolarity? Okay, something is going to happen. Now, before I talk about the osmolarity to this tubular lumen, we need to talk about the medullary interstitium, because outside this tubular lumen, there is osmolarity that is present here in the outside, or in the medullary interstitium. Again, in the medullary interstitium, there is already osmolarity. Okay, this osmolarity that is coming is the one that this osmolarity that is coming is going to be changing, but the outside osmolarity is already present. No wonder I'm going to be writing the values, but I'll talk about where this osmolarity is coming from in the medullary interstitium. So this one is going to start from 400, 400 moles per kg. Then we're going to have 600, 600. Then we're going to have 800. We're going to have 800. We're going to have 900. We're going to have 1000. And we're going to have 1200. Remember that this is mole per kg, so I'm not just putting the moles per kg, the units. So I'm going to have these numbers. These numbers, this osmolarity present in the medullary interstitium, I said I'll talk about where that number is coming from. Okay. Similarly, also on the other side. Okay, on the other side, because remember that this one is going like this. Okay. Also on this side, on the ascending limb, we're going to have also in the medullary interstitium the what? The osmolarity as well. Okay, it's the same osmolarity. Okay, so here, since there is 400, also somewhere here we're going to have what? 400. So let me just put somewhere here, we're going to have 400. Okay, 400, then 600 also here. Okay, then 800, just the same amount. We're going to have 900. Okay, we're going to have 1000. Okay, we're going to have 1000, then 1200. Okay, same amount of what? Osmolarity, this side and also the other side. Okay, now let's talk about what's going to happen if you come down here. Okay, this 300 mosmol, the moment it comes in this medullary part, okay, medullary part of this loop of Henle, something is going to happen. Okay, because you know that the body doesn't like two numbers that are different. They like something that is equal. So mean that if this is 300, it will try by all means that this 300 equates the what? The 400. Okay, so what I'm simply saying is that, so I'm going to use something now which is dangerous. I'm going to use this one. So if this now osmolarity present in the what? Proximal convoluted tuber immediately enters the what? The descending loop of Henle, which is the thin descending loop of Henle, then what's going to happen? This osmolarity is going to increase, so that it's going to increase to what? 400. So that it's what? Becomes equal with what? This one. Now, the question that you can ask is that, how is this number coming from 300 going to 400? How is it increasing to 400? What's happening? So what's happening is this. Remember what I said is that this descending limb is going to have what? Epithelial, specialized epithelial lining here. Okay, and these epithelial cells, they're going to have some certain transporters. Okay, some certain transporters for water. Okay, these they're going to have some certain transporters for what? For water, and these are what we call aquaporins. So they are going to have some certain transporters for water. Okay, these ones are going to have some certain transporters for water, like that. Okay, so which simply means that water will start moving. So let me use blue. Water will start moving out. So start moving through these transporters. Okay, this is water that is moving out. Again, water is moving out. Water is what? Moving out like this. So water is moving out of this interstitium, from this tubular lumen into the medullary what? Interstitium. Why is it moving out? So if water moves out alone, so we are saying water is going to move out alone, leaving out what? The solutes. Okay. So water is going to be leaving like that. Then you are going to leave out the what? The solutes. So the solutes are going to what? Remain here. The solutes are going to remain. Now remember that if you have lot of solid in a certain compartment and you have small amount of water, what's going to happen to concentration? Concentration is going up, meaning that the osmolarity also is going to go up. Okay, because water is the only one that is leaving. If water leaves and the solid are not leaving, the osmolarity is going to go up. So this is what's happening. For 300 to go to 400 means that the number of particles now are increasing. Well, how are they increasing? Because water is leaving out that part to become more concentrated. Okay, same applies here. It's the same thing that's going to happen. Same thing that's going to happen here. Here, if water leaves, we're going to have about, if this is 800, also here it will be 800. If this was 600, also here it will be 600. As it going deeper, this will be 900, 1000, up until it reaches what? 1200, like that. Because water is what? Living. That's the main objective. And if you go back to the major principle that we had we go back here. What did we say? In descending limb, water is permeable only. Why is it permeable? So that the concentration of the osmolarity now equals with what? The medullary osmolarity. That's what happens. That's a major important principle. Now, I talked about these transporters, which is aquaporin. Let me write them so that you don't forget. So these are what we call aquaporins. Aquaporins. But aquaporins type what? Type 1. You need to remember about this, okay? These are aquaporins type 1. Type 2, they are found in the collecting duct. Make sure that you remember. So aquaporins. That's why. Okay? Now we transit. We transit to another part, which is this side. Okay? We go to this side. Again, this side, here, the fluid is going to continue going like this. It's going to continue going like this, like that, with the 1200 osmolarity. Okay? But if it starts to go up like that, okay? Remember that here it is what? 1200. Okay, like that. But if it starts going up like that, okay? What's going to happen? What's going to happen? Okay? But before I proceed this side, we need to demarcate something important here. Okay? So remember that if you look at the medulla is going to have two important parts: the outer medulla and also the inner medulla. Okay? So what am I saying? If you look at the medulla, it has what? Two important parts. So it has what we call the inner medulla. So let us start with the outer medulla. So it has the outer medulla. So let us say M, and also inner medulla. Okay? Outer medulla and inner medulla. Why am I talking about this? Because if you talk about the demarcation of osmolarity, okay, the demarcation of osmolarity, if you look at 400 to 600, this happens in the what? 400 to 600. Okay, this happens, so this demarcation, 400 to 600 osmolarity, basically this happens in the what? Outer medulla. So take note of this. Outer medulla are going to find the osmolarity of about 400 to 600. Okay, but starting from 800 to 1200, that's in the inner medulla. So that simply tells us that as you're going deeper in the medulla, the osmolarity what? Increases. Why is the osmolarity increasing? Because water is leaving from the what? From the tubular lumen going to the interstitium. But I said in the medullary interstitium, the osmolarity is always there. The only one that is changing is this incoming filtrate. It's the one that is what? Increasing the osmolarity. But here it's always there. But I'll talk about where this 400, 600 in the medullary interstitium, where it's coming from. Okay, where are those osmotically active particles coming from? I'll talk about that. Okay, that's wait on that. So since we've understood about this, then we can move on to this part. Now, if you notice on the ascending limb, as you're going up, now the osmolarity inside the tubular lumen and also in the medullary interstitium is decreasing as you are going up. Okay, so as you are going up in the ascending, now it is decreasing. How? That's the question that you ask. Here, the how question, how is this increasing? We said water is moving out of the what? Tubular lumen going to the medullary interstitium. What about that? You go back to the principle, which is the ascending limb, water is impermeable, meaning that water is going to remain here. It's not the one that is moving out, but permeable to what? Sodium chloride. It is sodium chloride that is moving out, or the ions are the one that are going to be what? Moving out in the ascending what? Limb. Okay? In the ascending limb. So here we are going to have also specialized what? Cells, epithelial cells. Okay, specialized epithelial cells with brush borders in the what? Ascending what? Limb. So this 1200, for it to reach 1000, for it to reach 1000, okay, for it to reach 1000, I'm going to use black now. For it to reach 1000, Okay, there has to be something that is moving out. So now here, since water is not moving out, water is remaining here. Okay, now something is moving out. We are saying ions. So these cells, they are going to be getting sodium and chlorine, sodium ions and what? Chloride ions outside into the what? The medullary interstitium. Make sure that you understand there. Okay, the reason why now the tubular osmolarity is decreasing as you are going up in the ascending limb, because the ions are moving out of what? The tubular interstitium. If the ions are moving out outside the certain concentration, what do you think to the osmolarity? The osmolarity is going to be decreasing. Okay, so now to make things simple, so if you want to understand what I'm talking about, we said if you want to solve for osmolarity, we said we need to have what? The osmotically active particle. Okay, osmotically active particle. So let's just say particles over what? Water. Okay, so maybe this will be very simple to understand. Particles over water. Okay, so we're going to say particles. Now here in this situation, we are saying the particles are what? Are the one that are what? Going what? Outside. So mean that the particles here, they are leaving the what? The tubular interstitium. Mean that the particles are what? Decreasing. Then what about the water? Water is what? Just what? Constant. Water is just constant. It's not what? Moving out. Now if you look at what's happening is that the particles are what? Are decreasing, but water is remaining constant. The numerator here is decreasing, but the denominator is remaining constant. What do you think to the O over number or the result? The result is that now this, if you divide, now you're going to have what? Osmolarity, that's going to be what? Low. So the osmolarity is going to be decreasing because particles and osmolarity are direct proportional to one another. If you decrease the particles, mean that the osmolarity also is decreasing. So that's what is happening here. Okay, so I'm moving out the particles, which is sodium chloride. Okay, and you are decreasing the what? The osmolarity. So all in all, what you need to remember is that if water moving out of the what? Certain area, meaning that the osmolarity is going to what? Increase. If now the number of particles or the ions are moving out, meaning that the osmolarity is going to decrease. Okay, you need to remember about that. So that's the point that I needed you to understand. Now the question that we're going to ask is that this ascending limb is going to have what? Two parts. So this part where this sodium chloride is just moving out passively. So this is what we call, they are moving or using what we call passive diffusion. Okay, so these ions that are making sure that this osmolarity goes down, they are moving out via passive diffusion, and this part is what we call the thin. Okay, the thin ascending limb. Okay, this part, this is what we call the thin ascending limb, because the ascending limb is going to have two parts: the thin ascending limb and the thick ascending limb. Okay, as you can see, this is thin, and this part is very, very thin. And if you look at this part, that is the thick ascending limb. But in the thin ascending limb, the sodium chloride, they are going to be leaving, and they are going to be leaving out by passive diffusion from the what? Tubular lumen into the medullary interstitium. Okay, as this going and this number is going up like this, we're going to have 900, we're going to have 800, like that. Okay, now the moment you reach this thick ascending limb, now something is going to be happening here. Okay, because this has to be now be removed. Okay, up until it reaches the 300, the one that we started with. Okay, up until this osmolarity, tubular osmolarity, reaches the what? The 300. But for that to be possible, in the thick ascending limb, there are some specialized epithelial cells that are very, very important, and you need to understand about this. Okay, Even if you try to forget anything, but never forget about these specialized epithelial cells that are found in the thick ascending limb of the loop of Henle. Okay? These, what do you call these cells? What do you call these cells? These cells are called what? They are called specialized epithelial cells that are responsible for what? Making the medullary interstitium what? Osmolarity. I said I'll talk about where these numbers are coming from. They are coming from this cell. Okay? These are the cells or epithelial cells that are responsible for creating the what? Medullary interstitium osmolarity or medullary interstitium osmotic gradient. These are the ones that are responsible for these numbers in the medullary interstitium. But how do they do that? That's the question that you can ask. How do they do that? These epithelial cells, they've got these brush borders, but on these brush borders, they've got some certain transporters, okay? Some certain transporters. So let us represent this transporter, very important transporter that you need to remember. This important transporter. What is that transporter? So this transporter is called sodium potassium 2-chloride co-transporter. Sodium potassium 2-chloride co-transporter. Okay, co-transporter. Remember what I said in the previous videos? We said if you say co-transporter, means that all the ions are moving in the same direction, or all the molecules that are there on that particular transporter, they are moving in the same direction. Okay, so this is what we call sodium potassium 2-chloride co-transporter. It's a very important transporter. What does it do? What does it do? Because here we said we've got ions that are there. We're going to have the sodium. So here I'm going to represent with the red. So here, sodium will be taken from this part. Okay, sodium will be taken from this part and taken inside. Okay, by this transporter. Again, what else? Potassium will be taken, but just one potassium. Okay, and what else is going to be taken by that transporter? 2 chloride ions. So how many chloride ions are going to be taken up by that epithelial cells using that transporter? 2 chloride ions they are going to be transported into these epithelial cells. So we're going to have 2 chloride and you're going to have 1 potassium ion there. Okay, and that's why it is called sodium potassium 2-chloride co-transporter. Okay, now if this happens, now something important that's going to happen here. So now you need to ask a question to say, what makes this co-transporter or this transporter possible to start working? Because it doesn't just work on its own. Remember that all the co-transporters that we talked about, they used what we call secondary active transport. That's what we said. So remember, these epithelial cells, they are very specialized. So these epithelial cells, for them to make use of this sodium potassium 2-chloride co-transporter, they use something, because this has to use the secondary ATP. Okay, not direct ATP, but secondary ATP. But what do they do? If you remember, on the basal surface of these epithelial cells, we've got some certain important certain important transporters that uses ATP, which we call sodium potassium ATPase. So here on the back of these epithelial cells, they've got these transporters. So what do we call these transporters? Now I'm going to use red. We call this as sodium potassium ATPase. Remember about this. So why am I talking about this? Because these, they use what we call ATP for them to work. Okay, if they use ATP for them to work, what do they do? If you remember what they do, okay, I'll write them here so that you see what they do. So what do they do here? So these, they're going to make sure that they push sodium Okay, they are going to push 3 sodium ions outside the what? The cell, and they are going to make sure that they bring potassium. They're going to bring what? Potassium. Mean that they're going to bring about 2 potassium inside the what? The cell, but using what? ATP. So that's what they do. So sodium potassium ATPase type of transporters are going to be using ATP, and they're going to be transporting sodium outside the epithelial cells and the 2 potassium inside the what? This cell. But what is the main purpose of this? The main purpose of this is to make sure that they are going to decrease intracellular sodium. So that's the main goal of this ATPase. They want to make sure that inside this cell, this specialized epithelial cell, we have low, very little amount of what? Sodium ions. So that if there is very little sodium ions in this what? Epithelial cells, they are going to drive this sodium which was what? Outside. Because this sodium which is outside, it is what? It is a lot. If this sodium outside this cell, it is a lot, it will start moving into the what? Into this cell down its concentration gradient. That's what we call secondary active transport. So again, the goal of this sodium potassium ATPase is to make sure that you have very little sodium ions inside this cell. And if you look at outside these epithelial cells, we've got sodium outside there, but inside these cells, outside these cells, we've got higher amount of what? Sodium in this part. Then what's going to happen? Sodium ions will start rushing into this cell because there is no amount of sodium inside the cell down its concentration what? Gradient via this transporter which we call sodium potassium to chloride transporter. No wonder this transporter is going to use secondary, it's the type of transporter is what? Secondary active what? Transport. The type of transport that's going to be used by this transporter, which is sodium potassium to chloride transporter, it is secondary active transport. Okay, now since we know what's happening there, now another important thing that you need to understand: where are these ions that are coming inside this what? Cell going? Okay, where are they going? So we look at the most important what? Ions. Okay, this sodium that is coming in, where is it going? Okay, it has some, so as you can see, this sodium that is coming in is going to be used by this ATPase so that it can be pushed out, or it has some sodium channels that are here, or sodium gated channels that are here, then so that it can push this sodium outside like that. Okay, same applies to chloride. So chloride also is going to have some chloride channel, and you're going to push chloride ions outside the what? The epithelial cells into what? The medulla what? Interstitium. But what about potassium? Yes, potassium also is going to be pushed via the what? The potassium channels and outside the what? The medullary cavity. But something is going to happen to potassium apart from also going into the medullary interstitium there. So the potassium is more like a clever ion. Doesn't want to go into the medullary interstitium, but it wants to what? Go back into the what? Into the tubular lumen there. So this potassium, I'm going to remove this, doesn't want to go into the medullary interstitium. It will go back. Okay, it has to go back. Why? Because this potassium likes to help other what? Cells. So I'm saying I'm going to bring this potassium somewhere here. Okay. So I'm saying this potassium, okay, this potassium is friendly, meaning that it won't go out, but it want what? Go back into what? The interstitium. Why does it want to go back? Because it has to help other ions also to go into the what? Medullary interstitium. It's more like friendly. Okay, it wants to help other what? Ions also to go into the what? Medullary interstitium, moving from the what? This space. Okay, but what we call these channels where potassium is going to what? Use? We call it wrong potassium channels. Okay, so we call it wrong K. Okay, So Rom K, so mean that potassium ions are going to move out of this what epithelial cells into the tubular lumen via this Rom K potassium channels. Now why is it going there? Okay, remember that apart from sodium chloride, potassium, also there are other ions that are present. Other ions like what? Magnesium. Okay, there are also other ions like what? Calcium. Other ions like calcium ions. So these two important ions, they need to be also reabsorbed back into the what? Bloodstream or into the interstitium. But how is this going to be reabsorbed? Because these, they require potassium for them to be what? Reabsorbed, or for them to go from the tubular lumen into the what? Medullary interstitium. Okay, but they use a very important method. Okay, you've seen these two epithelial cells. Okay, so these are also epithelial cells. Okay, these are epithelial cells, but between these epithelial cells there is what we call tight junction, because remember, tight junction, they help to anchor the two epithelial cells together. Okay, so what are they going to do? So this potassium, it will make sure that this magnesium calcium passes through what? This space, okay, into the what? So that they can go into the medullary interstitium as well. So potassium is friendly. Also potassium is going to go as well via that road. What do we call that? That path, we call it paracellular. So what do we call this path? We call it what? Paracellular reabsorption. So paracellular reabsorption. Okay, paracellular reabsorption. So paracellular reabsorption basically is the ions, they are passing through what? Epithelial cells. Okay, through epithelial cells. Basically, it's going to pass through the what? The tight junction up to the what? Medullary interstitium, and they're going to be absorbed back into the what? Bloodstream, and that's what we call paracellular reabsorption. Another important concept that you need to understand about this. So remember, I said between two epithelial cells, there are what we call tight junctions. So, but I just want you to remember about the important tight junctions that are there, that are going to allow magnesium, calcium, and as well as what? Potassium to pass through. Okay, these are what we call Claudine proteins. So Claudine proteins, so we've got two types of Claudine proteins. We've got Claudine 16 and Claudine what? 19. So these are the proteins of the tight junction that are going to allow divalent what? Cations. So divalent cations mean that these ions with two positives, which is this one and this one. Okay, these are the proteins that are going to allow the movement of those ions via paracellular pathway. They are into the medullary interstitium and definitely into the bloodstream there. Okay, so that's what happens. Complex pathway. Okay, and if you look at this part again, if you go back to this part where potassium is going back into the tubular interstitium, and it's going to create something. As you can see, it's more like positive. Everything is positive here, and that's what we call positive luminal potential. So positive luminal potential is the one that's going to drive paracellular reabsorption. Okay, so potassium is going to provide positive luminal potential so that it drives all these ions via the paracellular reabsorption process. Okay, so those are the important concepts that you need to remember. Now, just to connect everything, we said that now, as you can see, sodium chloride and also other ions, they are going to be present in the medullary interstitium, making sure that we have this type of what? This type of osmolarity. Okay, you need to remember, I said to say in the beginning of the video, I said to say this 400 up to 1200 is coming somewhere, and it's coming from where these epithelial cells with these specialized what? Transporter, which is the sodium potassium 2 chloride transporter, because those ions, those are the ones that will be pushed in the medullary interstitium and contribute to the what? Medullary osmotic what? Gradient or medullary osmolarity. Okay, you need to understand about that. Now, last important point that we need to remember here is this one. Okay, last important point that we need to remember is talking about this part. So if you look at this, this is what we call the blood vessel, the capillary. Okay, the one that is also more like the loop of Henle is more like a U-shaped type of capillary as well. So I'm going to wrap around the what? The loop of Henle like that, okay? But they are almost like U-shaped as well, okay? So these capillaries, what do we call them? We call them what? The vasa recta, okay? So let us look at this one. Now, let us finish up whatever we're talking about in two various important processes that we need to remember because they're going to be examined during an exam, okay? So pay attention on this. So we're going to have two processes that we need to finish. We have the first one, which is what? Countercurrent multiplier. And the last one is what? The last one is which we call countercurrent exchanger. Okay, so two important processes that we need to cover here. Now let's start with the countercurrent multiplier. So we're going to break down this word into two so that we fully understand. So countercurrent multiplier. So we're going to have countercurrent. What does it mean, countercurrent? So countercurrent, remember that we had the descending limb of the loop of Henle and as well as the ascending limb of the loop of Henle. Okay, so remember that in the ascending or descending, the direction of the fluid was moving like that. Okay, and in the ascending, it was moving like what? Like this. Okay, as you can see, the direction of the movement of the fluid, it is opposite. So if it is opposite to one another in the descending as well as in the ascending, that's what we call counter current. Okay, now if you go to multiplier, that's from the word multiplier. Multiplying means that something is being multiplied. Something is what? Being multiplied. What is that? So if you remember, in the ascending limb, we had the thin ascending limb and the thick ascending limb. Now the thick ascending limb is the one that is responsible for multiplying some certain fraction of ions or osmolarity. Okay, very, very important point that you need to take note. Again, in the thick ascending limb is the one that is responsible for multiplying the osmolarity. What do I mean? So if you remember, in the thick ascending limb, we had specialized cells, epithelial cells, and they contain certain transporters. If you remember, we had sodium potassium 2 chloride cotransporter. Those, they got sodium, potassium, and chloride, and we added those into those cells, and again, ultimately, we added those ions into the what? Medullary interstitium. Okay, medullary interstitium, so that now we can have that number of osmolarity from 400 up to 1200. Okay, it's because of this pump. Okay, these cells. Now, where is this multiplication coming from? Okay, so multiplication is coming from this part. This part. If you remember, I said to say here we're going to have about 300 here, and also in the, if this is the medullary interstitium, also we're going to have about 300. Okay, but this part is going to further decrease into what? 200. If you try to subtract the 300, I mean the 200 and the 300, or the 300 and another medullary interstitium osmolarity, apart from 300, because it's not always 300, maybe it can be 301 like that. Okay, so mean that there will be a number, there will be a difference between this number and this number. there will be a difference. Okay, maybe there will be a difference of 10, maybe there will be a difference of what, 20. I'm just saying. All right, so that difference, if you try to subtract, basically it always favors the medullary interstitium. Okay, so this difference, if you try to subtract, let's say here we had about 310, and if you try to subtract 300 and 310, you're going to have a difference which is what, 10. That 10 meaning that it is being multiplied. How is it being multiplied? If every cycle, if this fluid is coming here removing 10, another fluid comes removes 10, meaning that 10 plus 10 is being multiplied. Okay, so being multiplied means that the osmolarity in the medullary interstitium is going to be multiplied so that at least it reaches that 1200. That's why it's called a multiplier, because the osmolarity in the medullary interstitium is going to be multiplied every cycle. Okay, every movement of that what, tubular fluid coming from the glomerulus up to the what, the distal covulated tuber, there will be that fraction. Okay, so that's what it means, this one, countercurrent multiplier. Okay, now let's go to countercurrent exchanger. You need to know the difference between the two. So countercurrent exchanger basically is going to be provided by these specialized what, blood vessels. These we call them vasa recta. Okay, these blood vessels or specialized what, capillaries, we call them vasa recta. We call them vasa recta. Okay, we call them vasa recta. These are very specialized capillaries. Now we need to remember that vasa recta, basically we can only find vasa recta in what type of nephron? Juxtamedullary nephron. So I said we can only find these in the what? Juxtamedullary what? Nephron. Okay, and these are the only nephrons that are responsible for concentrating what? Urine. In fact, they don't actually directly concentrate urine, but they indirectly help in the concentration of what? Urine. Because concentration of urine, basically, we'll talk about it in the collecting duct, where there will be use of what? Antidiuretic hormone, because it's the one that's going to be responsible for what? Concentrating urine. Okay? Either way, we know that this is now vasa recta, basically mostly found in the juxtamedullary nephron, and these are a few nephrons. They're about what? 15 to 20%. And the cortical nephron, if you remember, they can be found in the cortex there, and they've got what? The loop of Henle that is very, very short, okay, compared to these ones. Okay, but those cortical nephrons, they don't have the vasa recta. No wonder they are not responsible or helping in the concentrating of urine, because they don't have this. Now let us talk about this. You need to understand about this now. So this vasa recta is coming like that as well. Now we need to understand something here. From the ascending limb, so this is the ascending limb now. This part of the capillary of the vasa recta, which is associated with what? The ascending limb. What happens inside here? We need to talk about two important substances that are very, very important. Two important substances. So we need to talk about what? The sodium. Okay, very important. And we need to talk about what? Urea. And another one, let's talk about just water. Okay, yeah. So three important substances that we need to talk about. So this is urea. Okay, this is urea. So now, if you are moving down like that, what do you think is going to happen to these substances? Which one is going to move out and which one is not going to move out, in case you are asked? Okay, so what's going to happen here is that in vasa recta, if you look at water will start moving out as you are going down, like that. Okay, so water will start moving out like that as you are going down. As you are going down, water will be moving out of the vasa recta. Why? Because if you are going down, the osmolarity increases. Okay, because water likes to go where there is increased osmolarity. But these two substances are going to be left in the vasorector. They are not going to start moving out. That's what's going to happen. Okay? Now, the moment you hit the ascending part now, and this capillary, this vasorector that associated with the ascending the loop of Henle, what's going to happen? Let us write now here. So what's going to happen is that sodium, urea, okay, and water. What's going to happen? Now, on this point, sodium is going to be what? It's going to be put in the medullary interstitium. So sodium is going to be put in the medullary interstitium. So we're not putting sodium inside the tubular lumen. We're putting everything in the medullary interstitium here. So sodium will be put there, as well as what? The urea will be put where? There, in the medullary interstitium. Okay? But water is not going to be put in the what? Medullary interstitium. Why? Because water likes to go where there is higher osmolarity. But water, the reason why water is not going, because as you are going up, the osmolarity what? Decreases. And water doesn't like where there is what? Low osmolarity. If you remember, if you're going up, we're starting from 1200 up to somewhere 200. So water can go where there is low osmolarity. Okay? So this is what happens. So as you can see now, the reason why it is called countercurrent, again, countercurrent, what does it mean? It means countercurrent, it means what? The flow of what? Fluid. The flow of fluid or blood is what? Different. As you can see, the direction, this is going like this, and this one is going like that. The flow in that vasorector. Okay, it's going differently. Okay, so this is countercurrent. What about exchanger? What does it mean? Exchanger means that we're exchanging things. Here, we're putting water inside here, okay, in the medullary interstitium. But here now, we're putting sodium. It's more like we're exchanging, okay? Here initially we're putting water, but here we're putting what? Sodium. And water is not what? Added there. Okay? It's a very, very important concept. So we're exchanging things. So that's why it's called an exchanger. Multiplier, I talked about it. Alright? Now, why is this vasorector important? I said it is responsible for concentrating what? Urine. And remember, this mostly happens in the what? Inner medulla. Mostly inner medulla, if you remember. So inner medulla is the one that's going to make sure that you're going to have these processes happening. Okay? Because in the inner medulla, that's why you're going to have high concentration, high osmolarity, about 1200. Okay? So that's a very, very important concept that you need to understand. Another important point that you need to understand is that the vasorector is not only sodium that is responsible for what, like concentrating urine or helping the concentrating of urine. Also, urea is important. And when it has to be reabsorbed into the medullary interstitium. If you remember, we talked about in the proximal convoluted tubule that urea is going to be reabsorbed back into the bloodstream, and this is the urea that was reabsorbed. Okay? If it is reabsorbed, it's going to be used in the what? Concentration of what? Urine. And we are going to connect everything that we've talked about with the exit in the collecting what? Duct. The full cycle. Alright? Now, this is where we end from, and make sure that you study more. All the best until next time.